All right, welcome, folks. You a little tired, Janelle? um, Today we have a lot to do, so I think it'll help the class to move a little faster. And of course, we only have one more day this week, so then we get a little 2-day weekend, and then we've got 3 days next week. Really 2, because Wednesday, of course, we'll be at home, and so that's kind of like a half day. So we're very, very close, guys. I just want to push you, keep you motivated, keep you encouraged. We're so close. We will have an exam on Tuesday, so we have that to look forward to. And after that, we will kind of coast into spring break. So I'm happy with where we are. I'm proud of you all. You've done really well in this unit. I just want to remind you to make sure that you're getting those assignments done as well. Yeah, I will echo those sentiments. So I definitely agree that spring break cannot come soon enough, but we're a week away. A week from today, you all will not have school. You'll be at home relaxing, or maybe some of you will be traveling, or maybe some of you will be outside. Hopefully the weather will be nice. So we're almost there, but we still have work to do. All right, so today's lesson is going to be about sex-linked traits and something called the karyotype, which we'll talk more about. The objective is really the same thing that we've discussed for the last 3 or 4 days, but today we're going to focus solely on sex-linked traits. The essential question today: how can we predict inheritance patterns using known information about the parents? So this is going to take on another new meaning today when we take a look at these karyotypes. Okay. Oops. So it's March 25th, 2021. It's the 5th day of our 7th unit. So nothing to write on this slide, but just kind of a point of interest here. Of course, we've got 2 sex-linked chromosomes, 2 sex chromosomes. One is the X chromosome. The X chromosome contains between 800 and 900 genes. And on this X chromosome, there are 155 million nucleotides. So 155 million A's, T's, C's, and G's. That's a lot. Keep in mind, chromosomes are, of course, DNA that is just tightly wound around these proteins called histones, but that's a little bit above the level of this course. The Y chromosome is the other sex-linked chromosome, or sex chromosome. It's much smaller. It contains only 58 million base pairs and only 200 genes. One of the more important genes that it contains is called the SRY gene, and this leads to the development of male phenotypes. So the development of, it leads to your sex characteristics, the male sex characteristics: broader shoulders, a deeper voice, more body hair. So here's a size comparison of the two. The X chromosome is much larger than the Y chromosome, as we can see. And this is actually a 3D rendering of the two of them. So this is their relative size. So what do we think, based on what we just learned about? A person who was born with 2 X chromosomes, what would that person be considered anatomically? Good. Thank you, Abigail. A person who was born with 2 X chromosomes is considered to be born female. And so a person who was born male would be born with what? What do we think? There's still 2. So you still, just like the other traits we were talking about, you always get 2. Which 2 do you think? Yeah, in this case, you get 1 X and 1 Y. So it's a little bit different. You might think that you get 2 Ys if you're born male, but you actually get 1 X chromosome, and then additionally, you get 1 Y chromosome. So I do want you all to write that down. People who are born female have 2 X chromosomes. People who are born male have 1 X chromosome and 1 Y chromosome. Now, keep in mind, folks, we have talked about this in the past. Sex and gender are 2 different things. So just because, and I'm trying to be intentional with my language here, I'm saying people who are born female. And in fact, you might even say people who are assigned female at birth. But because we're talking about genetics here, I'm choosing to use the language born female. And then also born male. Of course, this does not perfectly correlate with gender. Gender is considered to be a social construct that considers different personality traits, um, different socially constructed characteristics. When we're talking about sex, we're talking about how a person is genetically constructed, how a person is hormonally constructed, and how a person is anatomically constructed. So we're not really taking into account their chosen characteristics, personality traits, sexuality. That's not coming into play here. Okay. All of the sex-linked traits are found on the X chromosome. And because males only have 1 X chromosome, they are far more likely to actually express these traits. And we're going to take a look at why that is in a way that might make a little bit more sense to you all. But I do want you to write down what you're seeing today that's highlighted in yellow or in green if your preferred language is Spanish. Sex-linked traits are found on the X chromosome. Because males only have 1 X chromosome, they are more likely to express these traits. Okay, we're good. So here are some common sex-linked traits. Male pattern baldness is a sex-linked trait. Color blindness, muscular dystrophy are also sex-linked traits. There are many, many sex-linked traits. Okay, that's why the X chromosome is so large. That's why it has 800 to 900 genes. Those genes, of course, code for proteins. Those proteins, when present and when absent, might give you different outcomes. Okay, so this is a cool chart, and I do want you to write this chart down. So before I even reveal what's in the chart, why don't you go ahead and draw it? As you can see, we've got 6 rows and 3 columns. That first column has the 2 sex, it'll show the sex chromosomes, and it'll also show the 3 genotypes. We're going to have a column for female and a column for male. And in this example, we are going to be talking about a trait. We'll use the letter B, as in boy or brother or basketball or Balenciaga or any number of things. Okay. So of course the female sex chromosomes are XX and the male sex chromosomes are XY. Just go ahead and write that into the chart. I know you already wrote it. Hopefully you already wrote it, but write it into this table. So what do we think? What do we think the homozygous dominant genotype will look like for females if we're using the letter B? Do we have any thoughts about that? What will the homozygous dominant genotype look like for females? Any thoughts? Okay, this is what it looks like. X with a capital B superscript and another X with a capital B superscript. So we can see that it's homozygous because we have two of the same alleles, two capital Bs. And because they are capital, that's what makes them dominant. But because these are sex-linked traits, we have to actually place the allele on the X. We have to place it as a superscript or kind of like an exponent on the X. Again, these are sex-linked traits, and we only find sex-linked traits on the X chromosome. So for males who only have one X chromosome, they only get one B. It's just XB, capital B, Y. So based on that, what do we think the heterozygous genotype will look like for females? Would you say? That will be one of them. And it's heterozygous, so what do you think? What does hetero mean? Different, good. So there's one that's going to be a lowercase, and there's going to be one that is capital. Good. So for the females, the heterozygous genotype on a sex-linked trait is X capital B, X lowercase b. Notice that for males, there is no heterozygous genotype. That cannot exist because males only have one X chromosome, singular. So only one X chromosome for males, therefore they cannot be heterozygous. You need to have two to be heterozygous. They have to be two different things. Okay, and then last one, the homozygous recessive genotype. What do we think that will look like? And folks at home, you all can answer too.
Over lowercase b?
Yes, it's going to be one X with a lowercase b and another X with a lowercase b. And then for males, it's just one X with one lowercase b because they only have one X chromosome. All right, so I want to make sure that you all have written this table down. This is going to be super important. It's going to be very helpful to you as we go through some practice problems. This is the last type of inheritance that we will discuss. So we did complete dominance, we did incomplete dominance, we did codominance. We talked about multiple alleles as it relates to blood type. And today we're introducing sex-linked traits. So we've got those 5 types. Okay. So the females who have the heterozygous genotype, excuse me, X capital B, X lowercase b, those females are considered carriers of a trait. carriers are not affected by the trait, meaning that they won't get sick if it's a disorder. They don't actually have the disorder. But because they have the marker of that disorder, they do have that singular allele, they can pass that trait to their offspring. So we're going to throw this word carriers around a lot. So please be certain that you know what this word means. These people don't actually have the disorder or whatever the trait is. They don't have it. They're not affected by it, but they can pass it on because they do have one allele for it. Now notice that males cannot be carriers. Males cannot be carriers because they either have it or they don't. Okay. There are a lot of sex-linked recessive disorders. Sex-linked recessive disorders only affect homozygous recessive females, and they affect males with the recessive allele. So homozygous dominant females do not have sex-linked recessive disorders. Heterozygous females do not have sex-linked recessive disorders. They can be carriers. They are carriers for those disorders, but they don't have it. Homozygous dominant men, or I keep saying men, males, do not have these disorders. You can only have this disorder if you are homozygous recessive. Okay. All right, let's do a practice problem. Did you need more time, Sana? Okay. Let's try a practice problem. So it says hemophilia is a sex-linked recessive disorder that causes excessive bleeding. Let's use capital H for the dominant allele, and let's use a lowercase h for the recessive allele. All right, so with that information, I tried to, get kind of artsy, and on the outside of this square, we can see two things. We can see two eggs representing the mother, and we can see two sperm cells representing the father on the left side of the Punnett Square. So what information do we know about these parents? Can we pick out anything just by looking at their genotypes? Good, Sally, thank you. The mom is heterozygous. And what do we call a heterozygous female?
Carriers.
Thank you, Abina. So the mother is a carrier. She's heterozygous. That means that she does not actually, and thank you as well, Abigail, she does not actually have hemophilia. She's not a hemophiliac, but she can pass it on. Okay, and do we know anything about the father? Is there anything we should point out about him? Or about that person? Yes, he's homozygous dominant. So that means that he does not have the allele. He doesn't have hemophilia. He's healthy. Okay, so he doesn't have any chance of passing on hemophilia. The mother, however, does. So let's take a look at this Punnett square. What's going to go in the top left box? What's the genotype of the top left box? X capital H and X capital H. Yeah, good. So, excuse me. This would be a homozygous dominant. Well, you guys tell me, what do you know about this person? If that's the genotype. What is that person's phenotype? They do not have hemophilia. And what about their sex? What are they?
Female.
Yes, thanks, Sabina. Thanks, Abigail. And thanks, Sally. So this is a healthy female. They don't have hemophilia. Okay, what about the top right box? What's the genotype for the top right box? Ashley, do you know? Good. X capital H, X lowercase h, and what is that person's phenotype? You're right about that. What? They're a carrier. Excellent. So she actually is healthy, Sally. She doesn't have, or this person will not have hemophilia, but we can still consider them to be a carrier. They can pass it on. So they have the same genotype as their mother. Okay, what about the bottom left box? What are we going to see there? What genotype will we see in the bottom left? Diana, do you know? No? You don't get this? Okay, let's go ahead. So look at these arrows. We're going to take this one and swing it over. We're taking this one and we're dropping it down. So this is how we end up with, you see how this is really the same thing as this? This is that. Okay, so we need to do the same thing over here. But this time we're not in this row. We're in this row now. And we're not in this column, we're in this column. So what do you think it'll be? So we're going to drop this down. This comes down here, and this comes over here. All right, so when we're doing these Punnett squares, we're always dropping these on the top down, and we're moving these on the left side over. Okay, so X, H is going to go there, and Y will go to the right. So this is what you get. I want you to do this next one after I talk for a second. Okay, so we get X capital H Y in the bottom left. What phenotype do we get with that person? Good, thank you, Melissa. This is a healthy male. So this person has the same genotype as their father. Good. All right. Sayana. X lowercase h and y. And then what is this person's phenotype? Do you know, Lisa? Okay. Ashley, do you have any idea? Hemophilia. Yeah, so this person, Ashley was able to tell us that this person is male because, first of all, they have XY. So when we see XY, that means that the person is a male. And she was also able to tell us that this person has a lowercase h. That is the recessive allele. And that means that this person is not a carrier. Males cannot be carriers, Lisa. I just want to be clear about that. Males can't be carriers. They either have it or they don't. And in this case, unfortunately, this male does have hemophilia. So when we take a look at the results of this Punnett Square, what information do we know? What's the percent chance that these two people have a healthy child? What's the percent chance? Or let me say this, what's the percent chance that they do not have, or that they have a child who does not have hemophilia? Good, thank you, Abigail. There's a 75% chance that their child will not be hemophiliac. They could have a healthy female, a carrier female, or a healthy male, but there is a 25% chance of a carrier child. I'm sorry, of a hemophiliac child.
What does hemophiliac mean again?
It just means that it's a disorder that makes it difficult for your blood to clot. So even small cuts and bruises just end up bleeding a lot.
Oh, okay.
Okay. So this is one example. Any questions about this? Thank you for that question, Abina. Any other questions? Okay, we're good. Let's try another. Oh, let's practice. So baldness, we talked about that. This is a sex-linked recessive disorder. Let's represent the dominant allele with a capital B and the recessive allele for baldness with a lowercase b. Can you predict who will suffer from baldness? So we're looking at some different genotypes here. We're going to drop them into one of two buckets, not bald on the left, bald on the right. Okay, so X capital B, X lowercase b. Is that person bald or not bald? This is a recessive disorder. X capital B, X lowercase b. This person is not bald. Okay, so let me kind of maybe I didn't explain this well enough. Let me go back a while. Oops, too far. Here. So sex-linked recessive disorders, the only way you can be affected by these disorders is if you are a homozygous recessive female, meaning you're a female who has two recessive alleles. Or if you are a homozygous recessive male, meaning you're a male with only one recessive allele, because remember, males only have one X chromosome. Okay, so only people with those two genotypes will be affected by recessive disorders. So let's take another look at this. So that very first one we already did, that's not bald, because that person, what would that person be considered? What's the name for that person? X capital B, X lowercase b. What do we consider that person? Would you say? They're not bald. And what's another way we, yeah, thank you. Abigail sent it into the chat. That's a carrier female. So that we already know where we can place the carrier female once we get to it. Oops. Okay, this next one, X capital B, Y. What is that person? Yeah, that person's not bald either. Remember, the only way a male can be affected by one of these is if they have the recessive allele. So this person is not bald. X lowercase b, Y. What about that person? That person is bald. Good. They have the recessive allele, so they must be bald. X capital B, X capital B. Not bald. Good. X lowercase b, X lowercase b. That person is bald. We see two recessive alleles there. Good. We're starting to pick up on it. What about X lowercase b, Y lowercase b? Okay, a lot of you are jumping in saying bald. This is kind of a trick question. Can anybody tell me why? What's wrong with this? Right, exactly. This person is male, so we shouldn't have that second B there. We shouldn't have anything on the Y chromosome. The Y chromosome does not carry these sex-linked traits. Okay, so X lowercase b, Y lowercase b, that's not possible. Trick question on that one. Okay, then we already said that the carrier female is not bald. What about homozygous recessive? That person would be bald as well. Good. So both of those two genotypes that are in the bald bucket are homozygous recessive. That's a homozygous recessive male on the left and a homozygous recessive female on the right. All right, so anytime we see a dominant allele, as we can see in the not bald bucket, it's going to hide that recessive allele. It masks the recessive allele, okay? So we've got to be careful with that. All right, we're going to do some practice now. So same thing we were just talking about. Male pattern baldness is a sex-linked recessive trait. A female carrier of the trait mates with a male that has male pattern baldness. What percentage of their children will have this trait? Okay, so this is a Punnett Square problem. What is always the first step when we're doing Punnett Square problems? We need to make a key. Good. So in this key, we need to define what each of the alleles look like. Now, the keys are going to look a little bit different here than they have in the past. You can, at this point, choose whatever you want your key to look like, as well as, as long as it has the important information. So we need to define what each allele codes for, and then we need to find out information about the parents, and then we can go from there. So X capital B, what does that give you? What trait does that give you? And thank you, JT, for that answer, and Melissa. In this example, what does X capital B get you? Yes, good, Abigail. X capital B means healthy hair. You're not bald. And X lowercase b means what in that case? Yeah, you are bald. You have baldness. Or maybe not, not immediately, not early on in life, but you're more likely to develop baldness later in life. Okay. We also need to know information about the parents for these sex-linked problems. So we want to know the genotype for the mom. What information does it tell us in the question about the mom? What does it say? What did you say, Lisa? She's a carrier. We've got a female carrier. So what is the genotype for a carrier? And thank you, Melissa. What will be the genotype for a carrier? Thank you, Abigail. X capital B, X lowercase b. And then what do we know about the dad? What do we know about the father? Janelle, you said it. Yes, he's got this baldness. So what is his genotype in that case? Okay, now Melissa said the same thing as Janelle, but we need to be careful because dad is a male. So he doesn't have two X's.
What about, oh, yeah, never mind.
Go ahead and say it, Abina.
I was going to say the same thing Melissa said.
Okay, X lowercase b, thank you both, and Y. So we've got to be careful. People born male only have one X chromosome. Because he has the baldness, that means that he only has that recessive allele. Okay, excuse me. Let's do the Punnett Square now. So we're literally just going to put the parents' genotypes on the outside of the box. We're splitting their genotypes up. So there's mom. She is a carrier. She's got one dominant allele and one recessive allele. Dad is bald or will be soon, and he's got one recessive allele and one Y chromosome. One Y, thank you. Oops. Okay, we've got 4 of you in class today, so I'm going to each of you for a box. Are we ready? Ashley, I'm starting with you. What goes in this box? Good. Now, you said lowercase. That's fine. The word doesn't. Typically, we do the dominant. It really doesn't matter. Janelle, what's going on? That's lowercase. And we can literally follow these and see how we're moving these alleles over. Sayana, what would be in this box? X capital B Y. Good. And then Lisa, yeah. Lisa, can you finish this off? X lowercase b y. Good. Excellent job. All right. So, what percentage of their children are bald? Or have this baldness trait, I should say. %50. Thank you, Janelle. And can you just explain how you knew that? Excellent. So the 2 boxes on the right side of the Punnett Square, X lowercase b, and then X lowercase b, y, those 2 people have male pattern baldness. One of them is a female and one of them is a male. The 2 on the left side of the Punnett Square, X capital B, X lowercase b, that person is a carrier, so they don't actually have the trait. And then below that, X capital B, Y, that is a male who does not have the trait because he's got only one dominant allele. All right, so it's a 50% chance, 50% of their offspring, or it's a 50% chance that their offspring will have the trait. Good job. Okay. So for those of you here, I know my board, the resolution is not great, but can anybody not see the numbers in these images? What's the first number that you see, Lisa? What about you, Ashley? What's the next after that? Sienna, can you see the one after that? Can you see one, Janelle? Okay, good. So everybody here seems like they can see it. But there are some people who are colorblind, and some people are colorblind for specific hues. So they cannot see maybe red and green. They don't see the differences between that well, so they might not be able to see the number 7 or the number 12 or the number 9 very well. Some people struggle with blue and red, so they wouldn't be able to see 16 or 13 very well. So this is all because of a sex-linked recessive trait, believe it or not. It's called color blindness. So this question says a man with color blindness, and there's his genotype, marries a woman who is a carrier for color blindness. What is the likelihood they will have specifically a son who is colorblind? So we need to do the same thing. We need to make a key. X capital N, what does that allele get you? What trait does that code for? Yeah, not colorblind. You have color sight. And then X lowercase n, what does that give you? Color blindness, good. And now we need to know information about the parents. So what do we know in the question about the mother? She's a carrier. Excellent. Thank you, Melissa. Thank you, Lisa. And what will her genotype be in that case? Good. X capital N, X lowercase n. And then we are already given dad's genotype in the problem, so we don't even really need to exert ourselves for that. Now we make the Punnett square. We split up the genotypes on the outside of the box. I'm going to put mom on top again. I'm using these colors. Of course, men can use the color pink, and of course, women can use the color blue. So I'm not trying to make a political statement about that. I'm just splitting it up there. Okay. And now we're going to fill out these boxes again. I'm going to ask my 4 people who are in class with me to help me fill these out. Who did I finish with? X capital, X lowercase. Good. And who's Sienna? We'll go to the next box. Excellent. Thank you. You got it. Janelle, X capital and Y. Good. And Ashley. Wonderful. Thank you all. Okay, now we've filled out the Punnett square, but we haven't yet answered the question. The question says, what is the likelihood they will have a son who is colorblind? Good. There's a 25% chance that they will have a male child who is colorblind. Only one of these boxes, we've got 2 boxes that could be male, but only one of them gives us a colorblind male. We've got 2 boxes that could be a colorblind child, but only one of them is also male. So it's a 25% chance. Good job. Okay, one more. If a woman is a carrier for muscular dystrophy, which is a recessive sex-linked trait, and the man has the disease, what is the probability that a female child would have the disease? Okay, so again, we make our key. I'm using the letter D for this example. Of course, the dominant allele gives a healthy is a healthy trait, and then the recessive allele gives you muscular dystrophy. Okay, what is mom's genotype? Thank you, Melissa. One dominant allele and one recessive allele because she's a carrier. She's heterozygous. And then what is dad's genotype? Good. X, lowercase d, Y. So he has this disease, this disorder. Punnett Square time. One more time. Does anybody think they can give me all four really quickly? Oscar. Who knows this box, Oscar? Who do you remember from biology? Yeah. It'll be X, X, and capital D, lowercase d. What about this one? So we drop this one down. It'll be X, lowercase d. And this one's going to get swung over lowercase d. Does that make sense? You got the pattern now? Okay, what's going to go? Okay. So you put the Y first, the order really doesn't matter, but typically that goes first. Good. And then what about here? Good. I'm such a good teacher, man. I'm just kidding. I didn't teach you. I had you for chemistry, right? Yeah, that was chemistry. Okay. All right, we filled out the Punnett square, but we have not yet answered the question. The question asks, what is the probability that a female child will have this disease? What's the answer? Yeah, who's that? Ashley? Thank you, Ashley. There's a 25% chance. Again, there's a 50% chance that they have a child with the disease, and there's a 50% chance that they have a female child. But there's only a 25% chance that they have both of those conditions, both a child who has the disease and who is also a female. Oh, it says colorblind. It should say who has muscular dystrophy. My apologies. Okay, we're not doing that quizzes. Okay, let's see how much you all remember. So here we're seeing this dramatic artistic image of the moments before fertilization. In reality, there would be hundreds, if not thousands, of sperm surrounding this egg, so it would be a lot more chaotic. But, and of course, millions of sperm are released, but only a portion, only a small portion of them actually make it far enough to get to the egg. But let's see what we still remember from this artistic image. How many chromosomes come from the sperm? How many chromosomes do? Yes, good. And what about from the egg? Yes, good. They both carry 23. So that means that a normal body cell has how many chromosomes? A healthy body cell. Good, 46, 46, excellent. So this is where we start this conversation about what are called karyotypes. And I do want you all to write this down. A karyotype is a map of an individual's 46 chromosomes. And they are arranged in their homologous pairs. So the pairs that are the same size get arranged next to one another. And these pairs also, not only are they the same size, but they carry the same genes. The first 22 pairs are called autosomes. They are your body chromosomes. They code for genes that have nothing to do with your sex. But that last pair, that 23rd pair, is your sex chromosomes. And of course, these determine your sex-linked traits, as well as your sex. Okay. So this is pretty cool. We can literally, get images of these chromosomes from the nucleus of a cell. Science has come a long way. But we can see that they're arranged from largest to smallest, right? The first pair are the longest chromosomes. They have the most genetic information. Then that 22nd pair is very, very small. All right, so would this person be a male or a female? Good, Abigail. Can you just explain how you knew that?
There's no Y chromosome.
Yeah, so Abigail was able to look at that very last pair, the 23rd pair. The 23rd pair show you the two sex chromosomes. This person has two sex chromosomes that are both X's. There is no Y. So we know that that means that they are a female. They have two X chromosomes. Good. Remember, XX equals female, XY equals male. So I want you to stop and think a little bit. Which parent determines the offspring's gender? The mother or the father?
The father?
Good, Abina. Can you explain that?
I just figured. I don't know how to explain. I just figured.
Okay. Ashley? Yes, it's because the fathers carry a Y chromosome. So if we go back and look at these Punnett squares that we were doing, the only way to get a Y is if it came from your father. And the only way to get a second X is also if it came from your father. Remember, mothers can only give two X's. So they will always give an X chromosome. Fathers have the option of giving one or the other. Now, of course, this is not a matter of choice. It's totally random. Every single sperm has a slightly unique composition of genes and chromosomes. One sperm might have a Y chromosome and the other one might have an X chromosome. It's a random process. But fathers are ultimately going to be the deciding parent in terms of the sex of the child. Good, Ashley, and good job of being as well. Okay, now we developed these karyotypes for a reason. It wasn't just that we wanted to look at people's chromosomes because we thought it was cool. We developed these karyotypes to be able to diagnose and potentially treat or avoid certain diseases. So we can look at these karyotypes and determine if they are abnormal by seeing if there's the wrong number of chromosomes. Abigail and Melissa and several others were able to tell me that the normal human cell should have 46 chromosomes. A person who has 47 or 45 or even 48, that person has an abnormal number of chromosomes and likely has some type of disorder. These disorders might appear in either the autosomes, so one of the first 22 pairs of chromosomes, but they can also be disorders in the sex chromosomes as well, the last 23rd pair. We're looking at a specific karyotype. Can anybody see anything that's wrong with this karyotype? Or anything that's abnormal? So go pair to pair. Go from the first pair to the second to the third, blah, 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 and see if you see one that is missing a chromosome or one that has an extra chromosome.
Number 21.
Yes, good. Thank you, Abina. The 21st position. And is this person male or female?
Male.
Good. This is a male, and this male has an extra chromosome in the 21st position. This specific disorder is called trisomy 21. More commonly it's known as Down syndrome. So this person has Down syndrome. People who have Down syndrome have an extra chromosome on their 21st position. Okay. What about this one?
I think it's 20.
So 20, I can see why you think that because one of them looks really small, but that's just the way that it happened to be bent. It's the X. Yeah, thank you, Janelle. So if you look at the 23rd position, which are the 2 sex chromosomes, we're actually missing a chromosome there. So this person only has 45. This person only has 45 chromosomes instead of 46. This is a disorder called monosomy X. More commonly, it's known as Turner syndrome. So this affects people who are born female. They end up having webbed necks. So their necks they look to be wider because the skin is kind of more connected to their shoulder. They've got broader chests. They can have some developmental disorders as well. Oftentimes they have reproductive issues. They're typically shorter in stature. Let's look at it. So we've talked about this before, but I want to see how well your memory is. We talked about 5 specific sources of genetic variation. Here are 4 of the 5. One of them is the cause of these chromosomal abnormalities. Does anybody remember which one it is? And we can see that karyotype on the left has all kinds of abnormalities. You might think mutation. That's what you might think, but it's not a cause of these abnormalities. You might think crossing over, but that's not it either. So now we're down to either C or D. Yeah, thank you, Abigail. It is non-disjunction. So non-disjunction is when during meiosis, and I probably should have included an image, so my apologies, but during meiosis, the chromosomes don't separate properly. And so one of the cells, one of the 4 gametes that is produced as a result of meiosis, one of the 4 sex cells, is going to get an extra chromosome, and one of them is going to get 1 fewer chromosome than it should. So this could take place oftentimes this takes place in sperm cells. So unfortunately, fathers are also more likely to be the cause of these chromosomal abnormalities. But one of the cells ends up getting an extra copy, which would lead to an offspring who has at least 47 chromosomes, or one of them might end up getting 1 fewer than they should, which leads to a person who has 45 chromosomes. So that process is called non-disjunction because the chromosomes are failing to separate. So here are some disorders that are caused by non-disjunction. There's a picture of Down syndrome, trisomy 21. There's Punnett syndrome, trisomy 13. That person has a cleft palate. Oftentimes, trisomy 13 can also lead to progressive blindness. Some muscular disorders as well that cause the hands to clench. We also know that there's this concept of intersex. Now, this was formerly known as hermaphroditism, but that is considered now a derogatory term. So we should not be using that term, even though it was once used by the medical community. It's now not seen as the polite thing to say. Now the medical community uses the term intersex as well. And these are people who have, for one reason or another, sometimes it could be caused by hormones, but oftentimes it is caused by abnormality different. Go ahead. Yes, go ahead. Oftentimes it is caused by chromosomal abnormalities, like Klinefelter syndrome, which is XXY, so you have an extra X chromosome, or Turner syndrome, which is that monosomy 23, which is just a singular X chromosome instead of XX or XY. Then there's also Edwards syndrome, which is trisomy 18. There's trisomy X as well, which would be XXX. So you also have an extra X chromosome in that case. And then there's, as we already discussed, Down syndrome. Okay, this is the last slide. I know we only have a couple minutes anyway. But how do we get these karyotypes? We do this process called amniocentesis. This is when a pregnant woman consults with her doctor. They might find during a regularly scheduled ultrasound that something is not developing properly. And so they choose to do an amniocentesis, which is using this really, really long needle. Okay. So amniocentesis is the use of this really long needle to actually puncture the placenta and draw out a little bit of the amniotic fluid. And, you know, remarkably, the amniotic fluid actually carries the genetic information of the child as well. So we can pull that out, we can analyze that amniotic fluid, and we can develop a karyotype of the child and see if they are carrying any chromosomal abnormalities. This is not a particularly painful procedure. It wouldn't be considered non-invasive, but it's minimally invasive. And like I said, it typically is only done when the doctor has a reason to believe that something is not developing properly within the child. Okay. So that was it. We do have 4 asynchronous assignments. There is an exit ticket today. I'm sorry I didn't leave you with enough time to do it, but you can take care of it during 4th block or at home or over the weekend. As long as it's done by Wednesday, March 31st, we are all good. We're basically at time, so you all can start to head out if you want to. Thank you for being here. Good job today. We've done excellent with this unit so far. I think you all have picked it up really well. And I will talk to you all tomorrow.